This is procedure 1 for exercise 1A on the microscope. You have a plate in your lab manual and you'll need to label the parts for it. You'll be responsible for those on the lab practical. So you just need to turn to page 1-1 in your lab manual. It's procedure 1. And all of the labels that I'm going to be going over in boldface print. So before we get started, the very last one, lamp, mark that out because this particular scope has a mirror. So I'm going to use a red pointer so it'll show up better on this black. And I'm going to go over the parts, and you'll probably need to pause it to label each one, so you can pause it till you get it labeled. So that's why I'm going to go through pretty rapidly, so just pause it when you need to. I may not necessarily do them in the order there, but you'll need to kind of mark them off to know when you've done them all. This first part from here to here is called the eyepiece. There's a lens here that you look through with your eye, obviously. It magnifies 10 times, so label this part eyepiece. This tube here is just a hollow tube, and this is the draw tube. Draw tube. So you'll pause it while you get it labeled. This larger part here on the main body, this is the body tube. Body tube. This large knob here is the course adjustment. When you crank it, it moves the body tube to make course adjustments. So course adjustment. Then to make fine adjustments, this is the fine adjustment. The chrome part here that rotates, the chrome part that's moving is the nose piece, rotary nose piece. The part that's moving, the chrome part, rotary nose piece. The objectives are lenses that magnify different magnifications. On the plate, there's only 2 there, and so the smaller one, label low power objective. The larger one, label high power objective. The smaller one, low power. The larger one, high power. The part right here that I'm gripping is the arm. Arm. The part that's sitting on the table is the base. The base. Even though it's molded on the same piece, this vertical part is the pillar. Pillar. There's a bolt that goes through the arm and the pillar, and that allows you to tilt the scope or incline it. Now you may have to draw this in where you can see it because it's black on black and it doesn't show up much. So right about here with your pencil, draw in this where you can see it and label that bolt inclination joint. It's boldface there in your lab manual. Inclination joint. In zoology, I tell them this is where the little animals perform. So this flat plate here is the stage. The stage. These clips are the stage clips. They're for holding your slide in place so it doesn't slip around on you. On the left side here is the diaphragm, and on the plate it's turned to where there's a large hole where you can see the hole. It's not the hole, it's the metal around the hole. So label this round part that shows the hole in it, label that diaphragm. You have to look carefully on the plate, but there's a glass lens directly under the objective that condenses the light. And so label that little glass lens right there, condenser, condenser. With these scopes, your light source was going to be on the table, and this is obviously the mirror. Okay, I'm going to go over the parts again just to make sure we haven't left something off. So eyepiece, draw tube, body tube, coarse adjustment, fine adjustment, the chrome part, rotary nose piece, the small one, low power objective, the big one, high power objective, arm, stage, stage clip, condenser, diaphragm, pillar, inclination joint, base, and mirror. Now, you'll be making drawings on the other procedures with 3 different drawings on each page. And when you do the letter E, and I'll be showing you how to set up your page, you'll need to somewhere on there indicate what the letter E is used to demonstrate. So on your letter E procedure to be done later, somewhere on there, write these 2 terms down. It demonstrates magnification and inversion, I-N-V-E-R-S-I-O-N. That will be discussed when you view the recording on that. And on the one with the cotton fibers, it's the other procedure on exercise 1A. So on that drawing page, indicate that demonstrates depth of field, D-E-P-T-H, depth of field, F-I-E-L-D, meaning how shallow or how deep things are in focus. And that'll be demonstrated obviously when you see the slides, the recording for that. Okay, that should conclude everything you need for exercise 1A. Label the microscope, and you'll be getting another recording on the letter E and the cotton fibers. I'll be making a recording to show you how to set up the page. I described it, but I will set it up on the board, and I'll get back to that later.